this video is going to run you through parts of the mitosis lab. First thing that I wanted to mention as we started this one is I've provided some links to some other videos to help you understand mitosis. One of those was made by one of the other instructors, Ms. Naples, and she has a video that includes the lab models, which I don't have access to since I'm not on campus at this point. And so to see those models and be able to draw them, make sure you watch that video and then draw them in the spaces that are provided within your lab manual. Okay, what we're going to do is we're going to go ahead and move down to the part that starts talking after the models. You get circles to draw models, and then after that you get circles to draw cells that are in those stages. So what we normally do during this lab is you put slides under the microscope and you start to look for the various stages of mitosis. One as seen in a whitefish embryo. This is the whole, well, a slice through the whole whitefish embryo, and you can see it's made out of lots of different cells. If we blow that up to a higher power, you start to be able to tell what stage of the cell cycle some of those cells are in. So this is it on low power, and this is what that same thing looks like when you take it up to blue high power or high dry power. And so now you're starting to be able to see differences in the cells. Like that cell looks very different from this cell, which looks different from this cell. And so this is where we would start drawing what some of those cells look like and what the different stages are that they're in. We're going to be doing the same thing with plant cells. The stages, the same things are happening in plant and animal cells in the stages. They just look a tiny bit different, and I'll kind of talk about those differences as we start to look at those cells. So, when a cell is an interphase, there's an obvious nucleus, if it's a eukaryotic cell, of course. The outside of that nucleus is marked by the nuclear envelope, and that nuclear envelope basically obscures your vision of the DNA present within the nucleus, and so you can't see chromosomes. Now, the other issue here is the chromosomes are in this loose, thready substance. It's called chromatin, and chromatin is so thin that you can't see it under the microscope either. So for two reasons, you can't see the DNA present in the cells. If you see a cell with an obvious nucleus inside of it, in either animal or plant cells, then you know you've got to be looking at a cell that is in interphase. Now, animal cells, they do have nucleoli inside the nucleus, but it's a lot more difficult to see those. They don't stain super well in the animal cell, versus in plant cells, the nucleoli are very visible, and there can be any number of those present inside the cell. So if you can see a nucleus and a nucleolus or two, then again, you know that that cell is an interphase. The other thing is, since the cell spends about 90% of their life cycle in interphase, almost all the cells that you're going to see are going to be in this phase. So if you get lost or stuck, chances are the cell's probably in interphase. Next, what happens during prophase is the DNA that was in the form of chromatin starts to condense or supercoil into chromosomes. Chromosomes are such a tightly packed molecule of DNA plus proteins that you can actually see it using a standard light microscope. The nuclear envelope that had been the boundary to the nucleus also disintegrates. So for two reasons, you start to be able to see the DNA present inside of the cell. The nuclear envelope went away, and the DNA condensed into a shorter and fatter molecule that's easier to see with our microscope. I've always called prophase the plate of spaghetti phase, and that's because the DNA is visible, it's coiled, and it looks like a bunch of spaghetti noodles just thrown on a plate. There's no order to them, they're not lined up, they're not pulling apart, they're just thrown into the middle of a cell. So this is an animal cell in prophase, this is a plant cell in prophase. Next up comes metaphase. That meta actually means middle, and what's happening here is the chromosomes line up in the middle of a cell. This one is easier to recognize in an animal cell, and sorry, my picture didn't really have a very good cell in metaphase, so it's kind of blurry, but you can see the chromosomes lined up right there towards the middle of the cell. Now, one of the other differences between animal cells and plant cells is plant cell chromosomes tend to be much larger / longer than animal cell chromosomes. So these are lined up in the middle of a cell. It's just that they're so long, their tails stick out towards the sides of a cell. It's not a plate of spaghetti unless you were to take a fork and line the spaghetti up in the middle, but then you'd still have trails kind of coming off from that. So it usually confuses students, and they don't see the line that exists in a plant cell in metaphase, but that's just because those chromosomes are so much longer in the plant cell than they are in the animal cell. Next, the chromosomes that had existed in prophase and metaphase were what are known as duplicated chromosomes. That means that there are actually two chromatid held together at a structure that's called a centromere. In anaphase, what happens is the centromere gets ripped in half and one chromatid gets pulled one way and the other chromatid gets pulled in another direction. So what happens in anaphase is those chromatids separate towards opposite poles of the cell. What's pulling them is the spindle fibers, and you can't really see those in either of the pictures that I'm showing you. I've got some other pictures that I might show you here in just a moment, but you can see pretty clearly that the chromosomes are being pulled towards opposite sides of the cells in both of these cases. And so that's what a cell looks like in anaphase. During telophase, the nuclear envelope that had disintegrated back in prophase, it starts to rebuild itself and the DNA starts to unpack back into the chromatin. However, it's really hard to see those two things occurring. And so what we usually tell students to look for is actually cytokinesis, which is division of the cytoplasm. It's easier to see that because there's two distinct things that happens, one in animal cells and one in plant cells. Because animal cells do not have a cell wall, what essentially happens is these filaments start to pinch the cell in half. I want you to imagine that you were to put a belt on and then just tighten the belt until you split in half. That's what happens, and it creates this cleavage furrow, which is this indentation. That's where the belt is squeezing to rip these two cells in half. Because plant cells have a cell wall, they can't do that. The cell wall is too rigid. So instead of squeezing in half, what they do is they start to build a new cell wall in between the new nuclei that are starting to form. And so you get a plate that is new cell wall being formed in between those two. And I know it's kind of difficult to see, but right here towards the middle, there's a cell plate that's being formed. So in animal cells in telophase, you look for that cleavage furrow. In plant cells in telophase, you look for the cell plate that's being formed. From there, you get a bunch of questions from the lab manual. They show you this picture where they've pointed to a few cells using a couple of different colors of arrows. The green arrow, that's the plate of spaghetti that I was alluding to earlier. So we can see the chromosomes that are condensing. That means that this is a cell in prophase. It's really early in prophase, but it's still prophase. This is another one in prophase right here. The red arrow is pointing to a cell that's in interphase. You can see the nuclear envelope. You can see a nucleolus inside of there, but you can't really see the DNA. Here's another interphase, interphase, interphase. Again, most of the cells that are in this picture are going to be in interphase. The blue arrow, notice that we're pulling the chromatids apart towards opposite poles. So when we're pulling apart, that's anaphase. Stupid memory trick that Ms. Naples included was anaphase apart. The chromatids are being pulled apart, so the two A's go together. That's really the only decent anaphase that's on that slide. These are plant cells. You can tell because they do have a cell wall, and again, there's a really obvious nucleolus, which the animal cells tend to just not have. And since these are plant cells, they would develop a cell plate, not a cleavage furrow. Cleavage furrow and cell plates fully form at the end of telophase. Most cells are going to be in interphase because, again, that's where a cell spends most of its life cycle. What's the difference between plant and animal cells? That, again, goes back to plants get a cell plate because they have a cell wall. They can't just pinch in half. Animal cells can just pinch in half, so they get that cleavage furrow or indentation. I don't remember what the question for number 9 was asking you. Probably something about when do the chromosomes pull apart. That's anaphase, and from there you just have some multiple choice questions, and so you can make sure you have the right answers, and then that finishes all of those nice little questions. Now from here, I did want to show you, and of course I didn't leave it open, that would have made my life too simple. I wanted to show you some other pictures from a textbook that shows you what some cells can look like in the various stages. This textbook is not the one that we use for this class, but it had some really good pictures, and so I just wanted to show you what theirs look like. So plant cell, animal cell, and prophase. Again, this is what I called the plate of spaghetti phase, and so you can see the chromosomes. This was, again, just taken with a much better microscope than what we have in our labs, and so that's why it's so much clearer what's happening there. Metaphase, they line up in the middle. Now, one of the other reasons I wanted to show you these pictures is you can see the centrosome with the centrioles right in the middle, which those control the spindle fibers. You can see the spindle fibers as those kind of darker pink lines that are connecting to the chromosomes that are in the middle of the cell. I had a really crappy picture of Metaphase in the other slides, and so I wanted to show you what it could look like, and they can look really pretty. This one also just shows you how much longer those plant cell chromosomes are than the animal cell chromosomes in the middle there. Anaphase, this one's a later anaphase. The animal cell up over here is kind of an earlier one where they're just starting to separate, but remember anaphase, that's when we pull them apart. Telophase, you can see the cleavage plate as a kind of darker blue line right here in the middle. Did I say cleavage plate? Cell plate, if I did say cleavage plate, but you can see the cell plate being formed in the plant cell, and you can see that cleavage furrow over here in the animal cell. So it can be a really good idea to make some flashcards to help you study this. Basically just go onto Google and Google plant telophase, animal telophase, print the pictures out on one side of the card, and then label it with the stage that's happening in the middle, because what you're going to have to be able to do is recognize cells in the stages. What we usually do on practicals and quizzes is throw up a picture and say, name that stage, and so you need to be able to do that. Hopefully that helps you prepare for your quiz and your practical.